Welcome, Agent Gleevec. Your mission, should you choose to accept, is the treatment of chronic myeloid leukemia. It is essential that you target the ATP binding site of the BCR-ABL oncoprotein. The CML condition arises from a genetic abnormality known as the Philadelphia chromosome. This is caused by a reciprocal translocation between chromosomes 9 and 22, giving rise to a unique fusion gene, BCR-ABL. This hybrid gene is transcribed to form mRNA molecules that contain one of the two BCR-ABL junctions, E13A2 and E14A2. Both these molecules are translated into the P10 oncoprotein. The leukemogenic potential of P10 stems from the constitutive activation by juxtaposition with alien BCR sequences of the otherwise normally regulated tyrosine kinase activity of the BCR-ABL protein. Tyrosine kinases comprise enzymes that can transfer a phosphate group from ATP to a protein in a cell. PCR causes the oncoprotein to dimerize so that the two BCR-ABL molecules phosphorylate each other on tyrosine. It is the presence of the dimerization domain on the first exon of BCR and the lack of the ABL cap region that results in constitutive activation, resulting in uncontrolled signal transduction, and interactions with a range of effector proteins, causing deregulated cellular proliferation, increased adherence, and reduced apoptotic response to mutagenic stimuli. As the tyrosine kinase is the effector part of the oncoprotein, it was the logical target for inhibition. Gleevec is a targeted competitive inhibitor of ATP binding on BCR-ABL and is FDA approved for CML treatment as it inhibits proliferation and adherence as well as initiates apoptosis selectively in disease-causing BCR-ABL positive or leukemogenic cells. Although side effects are common, they are usually very mild and rarely lead to discontinuation of therapy. Whether the kinase is in the activated or deactivated state is determined by the conformation of the activation loop or A loop, comprised of residues 381 to 402 of the C lobe. In its active conformation, the A loop is open because it swings away from the catalytic center, making the kinase available for activation by phosphorylation. In the case of ABL, tyrosine 393 is phosphorylated and allows substrate binding. In the inactive state, tyrosine 393 is unphosphorylated and points towards the center of the kinase, mimicking a substrate by forming a hydrogen bond with asparagine 363. In this conformation, the mouth of the kinase is occluded. The catalytic binding site of the ABL kinase domain consists of the interface between the N lobe and the C lobe. Gleevec binds to ABL's distinct inactive conformation by contacting 21 residues. While binding with the A loop involves no structural changes, binding with the N lobe involves an induced fit mechanism that increases surface complementarity. This is characterized by the creation of a hydrophobic cage around Imatinib that results in further van der Waals interactions as well as a network of hydrogen bonds with not only the kinase domain but also surrounding water molecules, giving rise to an extremely tight fit that is responsible for the stability of the inactive state as well as Gleevec's high specificity. The development of resistance to the drug has resulted in resurgence of the disease in some patients. The primary mode of imatinib resistance occurs due to point mutations in the BCR-ABL ATP binding region. The most common mutation replaces the gatekeeper threonine residue at position 315 with isoleucine to create the mutant protein product. This sacrifices a favorable hydrogen bond between imatinib and threonine 315 and results in an unfavorable steric clash due to the bulky isoleucine side chain, resulting in hindering the drug's binding efficiency. Each exploiting distinct characteristics of the protein inhibitor binding mode, numerous approaches have been adopted in the discovery of new generation BCR-ABL inhibitors. In order to avoid mutations involving the inactive conformation, second-generation drugs such as Bristol-Myers Squibb's competitively inhibits the active conformation of the oncoprotein in the conserved region of the ATP binding site. Next, Novartis' nilotinib optimizes the inhibitor binding interactions in a cleft adjacent to the ATP binding site in order to treat CML. In terms of an overall outlook, although there are multiple drugs in the market today for CML, There still are numerous challenges such as resistance that will hopefully be resolved by the pipeline of future drugs.